Good afternoon, everyone. I'm Ed Winnick, Editor-in-Chief at GenomeWeb, and I'll be your moderator today for this Genome webinar. The title of today's webinar is Mining Genomic Literature for Variant Interpretation and Gene Panel Design, and is sponsored by Genomenon. Our panelists are Mark Keel, founder and CSO at Genomenon, Nicoletta Sideropoulos, Medical Director of Genomic Medicine in the Department of Pathology and Laboratory Medicine at the University of Vermont Health Network, David Seward, Assistant Professor of Pathology and Laboratory Medicine, College of Medicine at the University of Vermont, and Victor Weigman, Director of Translational Genomics at Q2 Solutions, a Quintiles Quest joint venture. You may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box on the upper right side of the control panel. When you click on Send To, please select All Panelists. We will ask the panelists questions after the presentations have concluded. And with that, I'll let Mark begin with an intro to Mastermind.
Thank you, Ed. Welcome and thank you to attendees. What I'm going to discuss today is what Genomenon has considered to be the Other Human Genome Project, which is to say the challenge of organizing genetic knowledge out of the medical literature. So the purpose of my portion of this webinar is to provide a brief overview of Mastermind, which is our genomic search engine for those of you who are not familiar with the tool yet, as well as showcase how many of our users have incorporated Mastermind into their workflow. So this is a diagram that's undoubtedly familiar to most of you in attendance. It's our conception of genomic diagnosis in both clinical and research workflows, going from sample to report or insight. The way that we've broken down this process is in a primary phase of analysis, which invokes a physical process of converting DNA into information, and then a secondary analysis process, which is predominantly digital, both of which have been reduced to practice, that then culminate in a tertiary analytic component, which involves interpretation. And this challenge requires manual curation of genetic variants and is currently the analytic bottleneck in genomic diagnosis. So this is the reason for that bottleneck. The challenge here is that genetic information retrieval from the literature is made difficult by the sheer quantity of sources available in the form of individual publications across many thousands of different publishers, as well as the complexity and richness of information that needs to be extracted and organized from these resources, that is to say, genetic and genomic information. So Mastermind has addressed this challenge by indexing the genomic literature. We have scanned the 29 million titles and abstracts that comprise the PubMed dataset, of which we've identified more than 5 million full-text articles for complete indexing that contain information relevant to genomics and genetics. From these full-text articles, we have extracted and annotated disease gene variant associations across the entire spectrum of human disease and every gene and every variant in the human genome. The diseases include both cancer and constitutional diseases, and the variants include coding as well as non-coding variants, and also incorporate when authors mention those variants at the cDNA or the protein level. And I'll emphasize here that because this is a highly automated process, our algorithms for extracting and organizing this information continue to evolve to maximize the sensitivity and specificity of our results, and we provide increasingly sophisticated algorithms for computing and presenting the relevance of this information. This is a screenshot of Mastermind, which is a web-based software. Searching by disease is performed by users who are interested in seeing what genes or gene variants have been associated with that disease in the literature, or conversely, searching by gene or gene variant in order for users to see what diseases those biomarkers have been associated with in the literature. And then the search can be refined according to clinical relevance depending on the circumstance that the patient is experiencing. Several of these examples will be showcased by David Seward that he performs with his colleagues at UVM, including using Mastermind to perform multi-parameter search. This is one example of one of those high-level searches at the gene level. In this case, this is DNMT3A, where Mastermind is returning all of the diseases that this gene has been associated with in accordance with the number of articles that make mention of this association. And this is meant to really exemplify the fact that Mastermind has a comprehensive understanding of all biomarker associations across a whole spectrum of human disease. So this data that I'm exemplifying here is useful for database assembly as well as downstream machine learning activities that we provide to a multitude of our clients. What I'm showcasing here is a typical association page that results from a more focused Mastermind search, where on the right is the article landscape for the results of that search, and on the left is the variant landscape that results from that focused search, and at the top are the ways to refine that search to maximize the relevancy of users' results. So in subsequent slides here, I'll just break out a couple of the feature sets in Mastermind. This set of features showcases the survey of the literature landscape that Mastermind provides to its users. On the left, you'll see what we've referred to as an article impact factor plot, where each of those circular icons is a single reference that results from a user search for disease and/or gene and variant. On the x-axis is the publication date of any one of those references. On the y-axis is the citation factor or impact of each of those journals, and the size of the icons for each of those references suggests the relevance of the content based on the user's search or search refinements. Over on the left is that information in list form at the top, with each reference called out by bibliographic information, including detailed information about the frequency of matches to a given search, in this case the gene search. And then below, to provide context for the relevance that Mastermind provides, we've evinced the user's search terms in context of full text sentence fragments where those terms were mentioned by authors. So this is useful for reference labs who are interpreting large gene panels or otherwise performing exome or genome sequencing interpretation activities. This next feature set is showcasing the gene variant landscape that Mastermind provides its users for in this example, for a particular gene search. On the left is an array of all of the mutations in this particular gene as they are populating the linear axis of the protein along the x-axis, juxtaposed next to the functional domains of the protein, where each of these bars represents an individual variant allele, and on the y-axis is a reflection of, on a log scale, the number of references that make mention of that variant in the literature. That information is then provided on the right in list form that can be filtered or searched or sorted. Mastermind also showcases whether those mentions of any particular variant come from the full text or otherwise from a high-level presentation of the information in the titles and abstracts. And this information usually reflects a 10 to 15-fold differential between information being presented in the full text as compared to the title and abstract information. Information like this can be examined in the software interface itself or otherwise in concert with working with Genominon's team. Users can be provided with batch consumed databases. One salient example is a recent use case for a pharmaceutical company in phase 3 FDA approval for a development of a drug to combat a specific disease indication. We were tasked with producing a comprehensive database of all variants for 150 genes relevant to that particular disease indication to provide a database of companion diagnostics for this pharmaceutical company. Other ways to automate Mastermind including examining the data through data feeds or APIs, or otherwise engaging Genominon in using Mastermind's services, including gene panel design services, an example use case that Victor Wegman from Q² will discuss later, as Genomenon and his group produced a gene panel design for acute myeloid leukemia. To better reify what Mastermind is able to do in the way of informing gene panel design, I invite attendees who are interested to visit the website below for a download of a white paper describing the Mastermind process for panel design. To put it simply, we've started with a particular disease indication, just as an example, pan-hematopoietic cancer diseases, including leukemias and lymphomas. We then turned our attention to Mastermind to extract, organize, and annotate information for more than 300 gene candidates, and then output in a database all of the evidence support from prioritized references comprising the totality of Mastermind's database. And lastly, before I turn it over to Ed, I'll just encourage any attendees who have not already registered their interest in receiving access to Mastermind free, that they can go to the website provided here below and sign up and begin using Mastermind immediately. So with that, I'll turn it back over to Ed.
Okay, thank you, Mark. As a reminder to our webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'd also like to ask attendees to take a moment after the webinar has ended to take our exit survey and give us your feedback. I will now turn it over to Nikki Sideropoulos. Nikki?
Hello, thank you, Ed, for the introduction. I'd like to start out giving, as the medical director of the Genomic Medicine program at the University of Vermont Health Network, giving an overview as to what our program is, what our network looks like, and what our practice in delivering clinical genomic laboratory services also looks like. So that way, how Genomenon and Mastermind were relevant to us can be generalized to your own practices accordingly. So with that, my personal background is I am trained as a doctor of medicine. I'm a board-certified pathologist across the subspecialties that are listed on the slide. But my practice is primarily a molecular genetic pathologist, and I function as the medical director of genomic medicine at our institution. I'm an assistant professor of pathology and laboratory medicine, and I'm also, through my practice of genomics, have practice as a physician informaticist. So in terms of what our practice and what our network looks like in Vermont, we made a decision as a leadership group in 2013 at the time when we were thinking about genomics to really skip over classic molecular genetic pathology as it was practiced before 2013, more as targeting single genes in a more limited scope, and moving, because we weren't doing that practice. And then in 2013, with the resources that we had in hand, we decided to proceed directly to developing a genomic medicine laboratory with, and it's CLIA licensed, CAP accredited, and we based on our resource evaluation and relatively low volume, we service the northern regions of New York State and Vermont. And so our population overall is a little over 1 million that we serve. And with a relatively low volume, it made a whole lot of sense for us, in order to get increasingly more information from limited tissue, to move towards next generation sequencing technology as our backbone, and using that on the wet bench. And then we partnered with Purion DX to use the Clinical Genomicist Workstation for our dry bench and then report generation. So our our core philosophies in developing this program, we did perform a thorough stakeholder evaluation and secured our resources. A business plan had been proposed and signed off, and we also integrated our philosophies into the departmental strategic plan. And so our vision at the University of Vermont Health Network in this department is to practice clinical genomic medicine for the aim of improving health and healthcare. And by doing so, obviously we generate a lot of data, a lot of biospecimens, and so developing these banks of data and fueling genomic translational research. So by our clinical practice, we're driving genomic value and implementation research. But underneath it all, we are extremely dedicated also to genomic education. So literacy around how and when to use genomic-based testing, and then how to apply the results in clinical practice. We made the decision also as a philosophy to really push ourselves to go beyond just developing just the test, if you will, just the genomic test, and really think about developing genomic care pathways. And so the way I think about developing a genomic care pathway is that it involves strategic integration of the best genomic technology with the people and processes beyond the laboratory to realize the promise of precision medicine for each unique patient. And so it is in our business plan to develop a care pathway to service oncology, pharmacogenomics, and inherited disease. So some of our implementation considerations, we took a lot of effort to engage with institutional leadership to make this a reality. The end users, especially the clinical end users in particular, who are the people who are using our results? Are we servicing their needs? Obviously, our aim is to give the best possible service to patients. And we also feel like we have an educational role and a proactive role in engaging with payers of all different types to ensure reimbursement. So we made the personal decision to heavily invest in physician-trained genomic medicine staff. We have 4 people who are signing out our genomic assays, if you will, the reports, 3 of which are board-certified molecular genetic pathologists, and 1 of which is a medical geneticist with a background in laboratory medicine. And so in developing our tests, assay design in particular, I think the content has to be biologically and clinically relevant to service the end users in their clinical practice as they deliver patient care. In addition, I mentioned, being able to interact with payers, educate them in terms of what is the clinical, how was this test built? What is the content of this test? How is it built? Why were these targets chosen? What is the clinical utility, for these targets, and why is this going to impact patient care? And in doing, I think, our group has done, we think about doing our due diligence in terms of performing adequate, not only database searching, but medical literature searching to support the targets of the assay. And you've heard that that's a service that Genomenon is offering, and you'll hear more about that later in the webinar. But also what became incredibly, we put a lot of effort and resource into the medical interpretation of the variants that come out of our test. And the gene panel solid tumor, which is the first test that we launched, the genes of which are listed on the screen, we target every exon across those genes. And as you can imagine, we are trying to perform medical interpretation, and so, you could imagine by doing that breadth of coverage across those genes, you get a lot of variants where it's not entirely clear, how you do an adequate medical interpretation. It's hard to find information on a lot of these, and I think we lock down every process in the clinical lab to the best of our ability with operating procedures and going by, being a CAP accredited lab. Everything is locked down. But what we realized is that and this is in talking to our colleagues nationally and internationally, the exercise of searching the medical literature to annotate the clinical relevance of variants is really, I think it's fair to say, it's not a process that's locked down in the lab. And so, Dr. Seward in our practice may search in a little bit of a different way than I will. And sometimes we interface and we say, geez, well, you found this article and I didn't. And how do we lock this down? So we really felt that Genomenon offered a solution where we could take it into our clinical practice and effectively standardize the way we were searching medical literature. And we also felt that, I think any pathologist or anyone who's signing out a report, if you're going to make a statement that a variant has not been described in the literature previously, certainly you want to make sure that's true. And so there's been many instances where variants have come up. We think that we haven't found anything, but then we use the Mastermind tool, and sure enough, we find an article out there, and we feel like we can more adequately stand behind our reports. And we feel like we've done our due diligence using this tool. So, that's an important consideration for us, as is scalability. Our volumes are increasing, the number of tests we're offering are increasing, and so scalability at the medical interpretation stage is a real challenge, I think, for all of us in this particular field. So with that, I will hand over the presentation to my colleague, Dr. David Seward. Thank you very much.
All right, thanks, Nikki. That was a great introduction. I also want to just pass along my thanks to Mark and Ed for the introductions earlier. So, like Nikki, my clinical work is focused on molecular genetic pathology, and I'm one of the people who signs out the assay that Nikki described here at UVM. And the goals I have for today are to basically give everyone a flavor for what using Mastermind in the context of a genomic sign-out is like in a step-by-step basis. And to accomplish this, I've picked a couple of cases I've signed out in the last 6 months or so that I think will help illustrate the benefits to using this software and how it has directly impacted my clinical sign-out in my work. So the structure I'm going to use to accomplish this, I'll first go over the case at a high level and explain some of the variants that we detected, and also then articulate the questions that I asked and addressed specifically using the Mastermind software, and then walk through screenshots of the actual searches I did to give you a sense for how this information is presented, and then where I think the utility exists as far as pointing to the appropriate medical literature which I can use to support the final reports and my communications with our oncologist. So both of these cases are non-small cell lung cancer. That is currently represents the highest volume at our practice as far as the samples that we receive to sequence. And in this first particular case, one of the two interesting variants we found was an activating, well-described KRAS variant, the G12C. So I'm not going to spend any time on searching for this variant because I'm sure anyone who's signed out NGS assays or any of the classic molecular tests probably has, by this point, a canned comment for the KRAS variants. But what was interesting about this case is in addition to the activating KRAS variant, we identified a variant in the CDKN2A locus, which encodes both P16 and P14. And this particular variant is predicted to be a missense variant at the 83rd position. So it's a missense variant histidine to tyrosine in P16. And I had not seen this variant before, and so I'm going to use this as a starting point for using the Mastermind software. So the first question I had, quite simply, was, has this variant been published in the literature, and does it have a functional impact? Now, as many of you are likely aware, there are lots of publicly available databases where you can determine whether a particular variant has been sequenced in a tumor before, but frequently those do not offer any functional interpretation. It's just simply whether it's been identified previously or not. And so my question around this particular variant was specifically, is there a functional impact which I should try and incorporate into my report and communicate to the oncologist? So the strategy that I used in this particular case as a first step was, as Mark described, I kept maximum sensitivity options open by searching for all diseases, and then I specifically searched for the CDKN2A gene locus and the H83Y variant. In addition, I added an additional filtering step, which I'll document on subsequent slides, that helped to narrow the focus of the search. So in this first window, here is the Mastermind landing page. As you can see, I've entered the CDKN2A gene and the variant of interest. And after that first search, there were a series of publications identified. I then used the filtering tool at the top of the page, and in this case, I used the mechanism of action because P16 is a tumor suppressor. I was specifically interested in whether this variant would lead to a loss of function, which are the category of alterations that we are interested in for tumor suppressor genes. And as you can see, as I selected the filter on the top left of these options, loss of function, there were 8 relevant publications. So those are indicated in this next slide. On the top right are the 8 publications, and on the bottom right are the sentence fragments. So as the way that I use this in my practice, when I get a small list like this, I'll start at the top, highlight that paper, the sentence fragments will populate in the bottom, and I basically just quickly peruse through those papers, scan the sentence fragments for verbiage that would indicate the answer to my question. So in this case, I'm looking for, does this H83Y alteration result in loss of function? And as I scrolled over the 2014 publication from OncoTarget, the sentence fragment populated and said, essentially, yes, this mutation leads to the inability of P16 or the CDKN2A, an inability to stop the cell cycle or cell cycle arrest. And so at this point, the way I use this in my practice is I will then download the full text article, read the relevant passages where this fragment was attained, so that I can understand the context and evaluate the evidence the particular publication is using to support their finding. But at this point, in this particular publication, I found that the evidence was strong, and I incorporated that information into my report. So that's really, though, just step one. So now what I've done is identified, yes, I have an activating KRAS mutation. In addition, I have an inactivating CDKN2A mutation. So the obvious next step is this relevant for tumor progression? Is it relevant for patient care? Will it affect therapy? Will it affect prognosis? And to address that using the same Mastermind software, I took advantage of the advanced search criteria, where I can essentially search the relevant literature for only those publications that address concomitantly or combined situations where we're studying or looking for KRAS and CDKN2A variants. And so in this particular scenario, showing again the main search page and then the advanced search options, and I've included both the KRAS gene and the CDKN2A gene, and then used a keyword combined. So I wanted to make this clear. At this point, there's still a little bit of art involved in the searching when you're doing these higher level searches. So I tried a few things. I tried combined, I tried concomitant, I found a few others. So sometimes you have to be a little bit creative, but in this case, using combined, I then came to a master list, which, as you may expect, given KRAS and CDKN2A are quite common, there was a lot of literature available. But I kept my focus to the upper right-hand corner of the article plot, because this is the area where these citations are the most recent, as well as the impact factor of the journals being high. And I realize I sound a little bit like an elitist when I say this, but I do pay attention to impact factor, because my assumption is that those publications are likely to have been vetted and reviewed to a high degree. I realize that's not always the case, but it is the way that I choose to attack these searches. And in so doing, I identified this Cell paper from 2015, which specifically addressed CDKN2A deletions in the context of tumors having activating KRAS mutations. And what this paper showed was that there was the real possibility that these particular tumors would be sensitive to checkpoint inhibitors, so concomitant therapy with CHEK1 inhibitors and NK2 inhibitors. So I incorporated that information into my final report and had a conversation with the oncologist about potential clinical trials for this patient. So to move on to a second example, and I realize I might be running a little bit over, so I'll try to move through this efficiently. The second case is also a non-small cell lung cancer case, and just to give you the setting here, this is a patient who at their initial diagnosis, which was a couple of years before this, was found to have an exon 19 deletion that was known to be sensitive to EGFR inhibitors. So this patient had been on an EGFR targeted inhibitor and then progressed. So as you might imagine, when we sequenced this tumor, the original exon 19 deletion was still identified, the L747T751 deletion, but in addition, they had acquired the common resistance mutation, the T790M. So again, here's a case where I see this frequently enough in my practice that I know what I'm going to say about this particular combination. But the interesting part about this case is, in addition to the T790M mutation in EGFR, this patient had a secondary mutation in the beta-catenin gene, the CTNNB1. This was an S37C missense mutation. And at the time that this case came to my desk, I hadn't seen this mutation before, so I used the same approach as we discussed in the previous case to determine whether this particular mutation was in the literature and whether there was a functional association. So my strategy, again, starting at the main page, was to maximize my sensitivity by searching in all diseases and then searching for the specific variant, the S37C in beta-catenin, and then filtering those results using keywords, and I'll show you how I did that. So again, the landing page, beta-catenin, S37C, there was a series of papers found, but then I moved into the function filter and selected mechanism as a way to focus on papers where the functional relevance of this mutation beta-catenin was described. And in so doing, I was able to find a paper from 2007 where, as again, I scanned the sentence fragments, it was very clear that this paper stated that the S37C mutation in beta-catenin led to between a 3- and 7-fold increase in transcriptional activation, suggesting there was a functional impact. So again, at this point, I file this reference away from my report, and I ask the next question, which is there a relevance for having these EGFR mutations and the resistance mutation T790M in concert with an activating beta-catenin mutation in this case, in non-small cell lung cancers specifically. So I utilized the advanced search options to look both for EGFR and beta-catenin in non-small cell lung cancer where the T790M mutation was present. So this is a much more refined search, and in performing this, I'm showing the article plot, which Mark described before, and in the upper right-hand corner, which is always a great quadrant to start in, is a recent paper in a high-impact journal. So this is Nature Genetics. It's highlighted here on the bottom right, talking about the evolution of clinical impact and co-occurring genetic alterations. And within this paper, they specifically address this phenomenon of activating beta-catenin mutations in the presence of EGFR mutations and the relationship therein. And I was able to read this paper and communicate the findings, which basically suggested that co-targeting EGFR mutations and beta-catenin mutations in concert may lead to better outcomes in this patient population. So that's all I have to say for those two cases. I hope I was able to communicate a flavor for the way that we utilize this tool in our day-to-day practice and some of the benefits it may provide. And I'd specifically like to mention what Nikki brought up during her talk, which is this ability to really feel as though we've done our due diligence with the literature searches and not just putting in random association words in Google or in some other PubMed tool. We feel that, in our experience, this particular tool has been useful in generating appropriate literature to support our reports in practice. So with that, I will hand it over to Victor, and thanks again.
Yeah, thanks, David. Wait for the slides to go up. Great. Yeah, thanks again for the opportunity to chat today. I'll go a little bit different from David and Nikki here and kind of talk about the upstream part of panels that would ultimately make it into the clinic and the pathology that might be following up. So for those who are not necessarily familiar with EA Genomics or Q-squared solutions, we are a genomic laboratory suited for providing panel-based or sequencing services across the clinical trial continuum. So while certainly a lot of these panels can be used in small academic medical centers for testing, we're using these in clinical trials to either in a pan-therapeutic case for a given cancer, match people to the right therapeutics based on their mutational landscape, or in pan-cancer if we're having a trial for a given therapy. So we're always being asked by clients who want to come and create panels with us, what's the right one? Should we use this vendor or not? You know, is it too big? Is it too large? How can we make it faster, cheaper, et cetera, et cetera? And the team that we built here specifically is geared for expertise in molecular biology, bioinformatic analyses, you know, kind of getting what variants or mutations are present in the system. And not all of us are oncology experts in all things. And so a lot of times when we're making panels from something we're not as familiar with, this becomes quite daunting as we are experts in many different things, but not necessarily maybe your cancer. So what I'm showing here is a kind of pipeline for looking at this kind of thing. And really I'm focusing on the front left part. Because that's where we're at right here, and I want to emphasize here kind of in step one, looking at research and the biomarker identification is really difficult. In some cases, if it's a genotyping panel and we know a couple of markers that we're going to be identifying if a patient should receive a therapy or not, that's pretty straightforward. It's well known. David commented a couple well-known markers here that would dictate some kind of action. But in a lot of cases, folks want to be able to drive panels that have multiple utility. And so the amount of time we lessen on the left-hand side of this spectrum for a trial, for us, the better. Because after we've done that and we've made the reagents and we actually start running the assay, doing the bioinformatics, those are things that kind of have a little bit more finite timelines. But narrowing down that panel is very difficult, and it's something we do get asked on occasion. So, from that, we embarked upon an exercise about early 2015 now to create our own comprehensive cancer panel. And while certainly around that time this was becoming pretty popular in other areas and other companies were coming out with this, based on our clinical trial focus, we wanted to hit every marker that we could that dictated some kind of trial enrollment or druggable marker. So great, we have some databases that we could look for that, but what became very difficult very quickly is how we wanted to gather all that information that we didn't know from what we had on a trial landscape. Biochemical pathways being a big feature of this kind of stuff, classic items that I would have done in my past bioinformatics life in graduate school to really kind of narrow down what this list would be. So me and another individual and some other folks sat in really for several months pounding away at, let's start with these markers that we know are actively using FDA drugs. Here are markers we know are in ones coming up in the pipeline. Well, that left us with a small list that we felt might be incomplete. So really a lot of cycles here of high-end PhD time to come up with a panel that we were really happy at the end would serve a lot of different use cases for us. But certainly going forward, every time a client wants us to do this, we can't charge for 2 PhDs 6 months of time. That becomes a really intractable problem. And so when we want to build a panel, and clients are coming to us for this, I kind of give a highlight here of wants and don't wants, right? They want to be able to, if the panel isn't known or vetted or a vendor already provided it for some reason, they want that to be turned around. In a lot of cases, because trials might be therapy specific, but they might actually be specific in just classifying a tumor or in looking at risk-based markers. So things that might dictate familial inheritance patterns. And so we get multivaried needs that this panel may want to do. And of course, cost becomes a feature because next-gen sequencing is still kind of costly in some cases. And customization. Oh, I want this size for a panel for a trial doing this, another size for a different panel. So again, lots of variables. So I kind of show here on the right what I've seen most historically that we perform in that panel, where the core is, okay, what are the guidelines? Can I run a panel that's going to dictate NCCN guidelines for me that I can make sure patient management is appropriately streamlined? And then, okay, which ones are going to dictate treatment? Other ones might be, okay, I want to classify the tumor. And it kind of expands and expands and expands as we get into this field. But we don't want it to take a long time. We can't afford the FTE months I talked about earlier. So what next? You know, a year and a half or so ago, we had a large push from multiple different clients to create a custom AML panel. And now, and Mark elucidated to the white paper here, in leukemia, and certainly now over the last 6 months, even a year, the amount of myeloid and AML panels have really exploded. So, you know, we were seeing the same question every other vendor was seeing around the mid to late 2015 time of, we really don't have a great way to genomically profile AML. Okay, when we received this from clients, we said, okay, cool, I'm familiar with NCCN, I can pull up what those markers are. That's great. And from from this particular version of NCCN, I've got, 7 genes. For our pharma clients and other folks that want to get to a treatment response, that becomes a little bit trickier. Familiar with TCGAs in the subtyping category, but that's a whole lot of genes that are individually mutated, and AML suffers from a lot of indication-specific or patient-specific mutations. So finding hotspots gets trickier. And as I mentioned earlier, clients were asking, panels weren't sufficient, they weren't large enough, we can't subtype, we can subtype. Okay, we want the test that we can also use to diagnose minimum residual disease. Okay, the list goes on and on. So that's when I contacted Genomic folks to kind of help with this process. And so, kind of to gloss over some arenas here, what I wanted to do is just in a demoing of the tool in an afternoon, I said, okay, cool. Well, how quickly can I potentially build a panel in doing this? And so I knew, because we already had some tests that we ran in our laboratory here for FLT3 and DMNT3A, I put those in. Some of those are pretty common, and as David was speaking about before, really I started on the top right, looking at things that are most recent and highest impact factor. And so, right away, and you can't really see with the size very large, but there was really the small dot here that was highest impact possible. I highlighted on this and found a publication for genomic classification and prognosis in AML. Fantastic, great. Also the same thing, looking at FLT3, you can see the dots in the very top right. Clicking on that led me to this PubMed paper as well, which is also genomic profiling and AML. When I actually search for these types of fields, like genomic profiling of AML, drug targets of AML, a lot of different kind of Google searches before, none of these publications came up in a couple different arenas. So that was really difficult to really get this. But as I put in my last bullet here, in less than a minute, I had 2 papers to pull on that had large swaths of gene lists, subtyping, markers, et cetera, and that's great. Of course, I can't necessarily mine and do this for every single marker I want to add. So in talking to Mark and the team at Genomic, I said, this is great. I found papers I want. I can already take a lot of markers here to put in a panel. How can I stretch this out and identify more items? The slide here kind of shows your typical one-tailed graph where you've got your biomarkers here. This is from the PubMed manual paper. Some are SNVs and indels, some are complex mutations, some are translocations, copy number losses, et cetera, and kind of split out among risk profiles for patients, which is, again, outstanding. That's what I'm looking for. I want a panel that can hopefully, for me, hit a lot of different kinds of classes of genomic variation, not just your SNVs and indels, because I want it to be kind of broadly used. So, you know, all right away, these papers were exactly what I was looking for, the specific markers to throw into the panel. Of course, that's great, but these markers are really tricky. I'm not a cytogeneticist. I don't necessarily speak all the same lingo of that. So in asking Mark, is there a way we can mine this to further identify goodies? And so we went in on a high level for marker class, saying, okay, I want to define those that are, first, the reason why I found these two to begin with is because I was looking for prognostic markers as the first request. So I had highlighted here the prognostic circle. And from that, I got, you know, genes when I was looking at FLT3, and the markers that were highlighted on the protein diagram were prognostic related. Going further, I could then filter those for things that are treatment related or diagnostic related. And so for me, that added that kind of variability, going back to the kind of concentric circle graph that showcased wide range of uses that a client might need. And I could start classifying, at least here by hand, markers and genes that are of interest. And that's great. So digging into the underlying database, how do I really start doing this kind of wholesale? And working with Mark and the team, we started doing so by providing a list of publications for me were most relevant for, you know, your prognostic markers, your diagnostic markers, your treatment markers specifically. And what we came back from the underlying engine within Mastermind was to actually create a prioritized list of individual markers. So this isn't just a gene. As you see here in the top left for SNVs with KIT, you're also seeing the biomarker, the D816V, as being, you know, highly active. You have other ones here in FLT3 and IDH, NPM1, things that I knew right away that circulated to the top, and specifically the markers, so that when if I really wanted to make a tiny panel, I could actually focus only on those genes should a client need something like that. But given something like AML, SNVs and indels are only a tiny part of the story. Knowing genomic rearrangements, fusions are a big part. BCR-ABL, of course, yep, certainly remembered that. I was familiar some with RUNX1, but not a whole lot. But we were able to pull out that information for fusions and copy numbers very quickly, which was awesome. And actually, going even further than that, and I didn't expand the figures here, but we're able to go back to those papers in a lot of cases, and the database here is actually identify the genomic coordinates of those things, which I need to create an enrichment panel. So kind of after doing this, going and following this concentric circle line, I took the union gene list from all these markers that were prioritized in publications that had associations to, in some cases, treatment, relationship to outcome, or diagnostic related. So really, is there a marker that's going to not just identify you as AML, but you as this particular, you know, subtype of AML, which we did. And then those that might be risk factors for clients that want to provide additional value for running in that trial. So we made a union list of about 120 genes that could be classified across these different biomarker classes, treatment, diagnostic, prognostic, or risk-based. So we can kind of mix and match with prioritization of the individual markers to provide a variable kind of panel design right away. And this whole process took, you know, a very short amount of time, a couple weeks, to have something I could go back to several clients and say, here's our basic design. Let's go ahead and go on with this process. Which is great for us because that's what we want. You don't want to wait a couple weeks to come into that, or more than a couple weeks to do that. You really want to start talking to folks about narrowing in that content. And that's been a hard conversation as well, unless you can prioritize based on these individual markers rank in publication and providing that evidence for their inclusion, which is great. So from that, to kind of wrap it up to allow some time for questions, you know, we are able to identify custom content. I'm not an expert in AML, not at all. I'm a solid tumor guy. But this was very encouraging for me in just a short couple minutes of an afternoon to pull out, you know, hundreds of individual protein changes or markers that I might be able to add. So that was awesome. The context of that gene, is it prognostic, is it diagnostic, is it treatment related, was also great. I mean, one thing, looking into those combination markers to therapeutics can also enhance this stuff that was mentioned earlier. Coming up this stuff quickly, again, very valuable, and after that screening is done, as I discussed, we're able to go back and say, here's your panel list. Okay, if we include these top 100 markers, not necessarily genes, but markers, your panel is going to be this size. We can run this many samples at a time. It's going to cost this much, and it allows us to start looking at some economic and actionable decision criteria for creating that panel, which is really great. From that, I'll wrap it up, and we can start taking some extra time to answer some of you guys' questions. And thank everybody for joining.
Okay, thank you, Victor. As a reminder to our webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'd like to ask the attendees to take a moment after the webinar is ended to take our exit survey and give us your feedback. We will now start the Q&A portion of the webinar. The first question is for Mark, and Mark, a participant would like to know how often the publications database on Mastermind is updated.
Thank you, Ed. That's a great question. I apologize for neglecting to mention that during my portion, but Mastermind is updated on a weekly basis for both all titles and abstracts that have been published in that interval week, as well as any of those titles and abstracts at the full text level that contain genetic or genomic content. So the short answer there is every week.
Okay, thank you. Mark, the next one also is for you, and one of the participants would like to know, is it possible to search the results by other keywords, such as treatment keywords?
Excellent question, and so what Drs. Seward and Wegman exemplified was different approaches to prioritizing, filtering, and augmenting the relevance of search results. The typical lines of refinement include diagnostic, prognostic, and therapeutic significance. There are ways to provide user input to refine that search that are available in the professional edition of Mastermind, and it's an important place for me to also emphasize that as we're continuing to refine our algorithms and ways of parsing the data, for those of you who are interested in constitutional genomics, we're promulgating use of ACMG classification criteria into our categorizations based on keywords. So short answer is free text is available in the professional edition, and there's a battery of additional categories and key terms that are currently available in the professional edition, as well as in the future state ACMG classification criteria.
Okay, thanks, Mark. We have a question for Nicoletta. Do you have any thoughts on how the information from Mastermind could be used to help educate physicians on genomic medicine?
How the information from Mastermind can be used to educate?
Yes.
Yes. So obviously, in listening to what David Seward presented, we are searching for medical literature that's pertinent to the clinical relevance of variants that we identify via our testing. What we end up doing is then reviewing that literature, reviewing that content in the relevant articles that we identify via Mastermind, and we summarize that information in the clinical report. A lot of times there are discussions with the clinicians on the other end receiving these reports around exactly what information we are entering as text into, and as a variant classification into the report. So there's education at that level, and also, you know, we're at an academic medical center, so it's not unusual at all for residents, medical students, fellows to be on service with us, and you know, we are very careful to include PubMed IDs in our interpretations to, so that way the end user can always have the option to go back to the primary literature and cross-check us. So I think that also serves as a very rich database of information that we primarily get by using Mastermind for all sorts of levels of you know, medical clinical expertise to go back to, read primary literature, get educated on that, learn how a molecular genetic pathologist or a medical geneticist, in fact, interprets the primary literature identified via Mastermind, and then makes a you know, laboratory medicine decision around how to import that into a clinical report. So hopefully that sheds some light on you know, for that question.
Okay, thank you, Nikki. Mark, we have another question for you. Is there any chance that rather than typing in individual variants, users could upload text files or Excel files with all the variants from a single patient at once, along with relevant information like disease?
Great question. Mastermind does afford those automation capabilities. At present, we have a VCF file annotation process for every line item of data, which is just a genetic variant. The VCF processing will annotate those variants and the numbers of articles that mention those variants, and then in an enhanced version of that VCF annotation process, there are ways that APIs can be utilized or VCF annotation can be modified to include suspected diseases for the given clinical circumstance. So short answer there is batch VCF processing is probably the easiest way to get that information, if not through API data feeds.
Okay, thanks, Mark. We have a question actually now for, it could be for Victor, but also we'd like David and Nikki to weigh in if they'd like to. Victor, let's start with you first with this one. Can you quantify the return on investment or time savings that you achieved using Mastermind?
Sure. So for us in that AML example, we were able to churn out, again, with Mark's help kind of probing the back end a bit, about 2 weeks to, well, really 1 week to generate the full kind of union list, and that is kind of actual time. For me and the other individual at EA, that might have been a few hours back and forth with Mark discussing, you know, and kind of mining ourselves. So on my end, you know, I would say 8 hours, 1 PhD time to help filter and prioritize different lists based on those interaction items. Within that week, we had the master list, and the other week was spent creating a way to rank the markers so that when we gave the clients who had an interest of treatment, then we could automatically, very quickly, give them a marker list ranked by treatment or ranked by prognostic. So the other week was spent creating the panel in such a way that clients who were asking could easily shift the markers in a way that suited their needs. So really, you know, it might have been 30, 40 hours of my guy's time versus something where it would have been my guy's time out for a month or so real time. So for me, that's pretty quantifiable. And since we get multiple different requests, I don't necessarily have folks I can take out for that long.
Yeah, so I can.
Go ahead, David.
So I can address this from our side. So when we were first introduced to the software, we had already been practicing without it for some time, and so I'd done a numerous side-by-sides where I'd either start with my the way that I'd been searching prior to using Mastermind, and then looking for using Mastermind, using the same search, and vice versa, trying to compare what articles I found and the time it took me to find those. Now, the difficulty in trying to answer the question specifically about time quantity has to do with what I refer to as my comfort level with my report. So prior to being convinced that Mastermind its sensitivity was appropriate, and before using Mastermind, I frequently would continue to search up to the point where the report basically was due and I needed to get it out, and that was the timeline. So however long I needed to be reading, because what you'll find with a lot of these variants is there is a massive amount of literature, and so if you don't try to narrow your search in a rational way, you can be reading papers for the rest of your career on some of these topics, and they're continually being updated. So where I found it to be particularly advantageous was the sense of search completion that I had done. So after perusing the top hits, doing the filtering in Mastermind, I didn't have that sense that I'd missed something, because in my analysis, whatever I found doing my searches through Google Scholar or any of the other search engines out there, those inevitably were captured by my searches in Mastermind. So it's not so much about time quantity as it is about the feeling that my search has been complete. If that doesn't make sense, I can try and you can re-ask a more specific question and I can get to it. But for me, it's about the sense of having done my due diligence, and that is in my side-by-side comparisons that has been achieved with Mastermind.
Okay, thanks, David. Nikki, did you want to chime in on that question?
I do think that for me, as a medical director, being able to have a sense of confidence that we have standardized our process of how we're searching the literature has been a relief. I think anytime you can lock something down and feel like there's consistency across the group, across how we're doing our medical interpretation, that's an invaluable thing to have. So I'll keep it short. I understand we're up against the hour, but I did want to make that final point.
Okay, thanks, Nikki. And that is all the time that we have today for the webinar. We'd like to thank our panelists, Mark Keel, Nikki Sideropoulos, David Seward, and Victor Weigman, and our sponsor, Genomenon. If we didn't have time to answer your question, we will try to have the panelists answer them directly afterwards. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.